Welcome to chapter 17. Now we get to talk about nucleic acids and protein synthesis. So histology, you guys know, is when technicians take a look at slides that are of tissue and they're stained with different stains. So histotech studies the microscopic makeups of tissues, cells, and bodily fluids with the purpose of detecting and identifying the presence of a specific disease. They determine blood types and the concentrations of drugs as well as other substances in the blood. So, when we talk about nucleic acid and DNA, right, we kind of have to understand what the basis is before we can move on and actually understand what's going on. So, the general structure of a nucleotide contains a nitrogen-containing base, a sugar, and a phosphate group. So, you can see the base, the sugar, and a phosphate group. And this is called the backbone. Okay, so in DNA, the purine bases are adenine, guanine, cytosine, and thymine. Where RNA is a little bit of a diva, right? And it's like, I am way too important for T's, so you have to use U's. And so RNA contains the same, except for the thymine, T, is going to be replaced by uracils, or U's. So we're going to learn how to describe the different bases and the ribose sugars that make up the nucleic acids, DNA, and RNA. So when we're talking about bases, the nitrogen-containing bases in a nucleic acid, they're derivatives of pyrimidine or purine. So your pyrimidine is here, and it's a nitrogenous ring, where your purine would be two nitrogenous rings together. In DNA, the purine bases with double rings are adenine and guanine, and the pyrimidine bases with single rings are cytosine and thymine. So this is going to be very useful when we talk about what can bind with what. A will always bind with T, C will always bind with G. So it's one from the pyrimidine group and one from the purine group. And again, RNA is going to have the same bases, except for thymine is going to be replaced by uracil. So when looking at the DNA again is going to contain bases A, G, C, and T, where RNA is that little bit of a diva, and it's going to have A, G, C, and U. So again, the pyrimidines are going to be cytosine, thymine, and uracil, which is in the DNA, RNA, or the RNA diva, right? And so that's only going to be an RNA only, where the adenine and the guanine, these are purines, and these are in both DNA and RNA. So talking about what pentose sugars are, the pentose or 5-carbon sugar in RNA is a ribose, where in DNA it's a deoxyribose with no oxygen, the no oxygen atom on carbon 2. And it has carbon atoms that are numbered with primers to distinguish them between the atoms and the bases. Okay? And so remember, the RNA is just the one ribose, and DNA is the deoxyribose, and that lends us to the D. So a nucleoside, this is going to be a combination of a sugar and a base, and it's produced when the nitrogen atom in a pyrimidine or a purine base forms an N-glycosidic bond to the carbon 1 of sugar, and this would either be ribose or deoxyribose. And it's named by changing the base ending to either osine for purines and iodine for pyrimidines. So let's look at an example. Adenine and ribose form a nucleoside called adenosine. Okay, so you have your ribose and you have your adenine, and when they combine using that N-glycosidic bond, the name now is going to be adenosine. Well, nucleotides, these are nucleosides in which phosphate groups bond to the OH, or that hydroxyl group, on the carbon 5 of ribose, or the deoxyribose, to produce a phosphate ester. And they are not formed when other hydroxyl groups on ribose form phosphate esters, only on the 5' monophosphate nucleotides that are found in RNA and DNA. So those nucleotides, they are RNA, and they're identical to those of DNA, except for, remember, the DNA is the sugar deoxyribose, and the deoxythymide replaces uridine. Okay, so DNA has T's, where RNAs has U's. Okay, so looking at the components of DNA and RNA, well, DNA has A, G, C, and T, The sugar is a deoxyribose, and the nucleoside is a base plus a deoxyribose. The nucleotide is a base plus the deoxyribose and a phosphate, and then the nucleic acid would be a polymer of a deoxyribose nucleotides. Where RNA, remember, it's our little bit of a diva, and that's going to have our A, G, C, and U, and our sugar is going to be the ribose. We have the base for the nucleosides plus a ribose. For the nucleotide, it's going to be a base, a ribose, and a phosphate. And for the nucleic acid, it's going to be a polymer of ribose and nucleotides. So in summary, how do we name nucleosides and nucleotides? The names of DNA nucleosides add a deoxy to the beginning of their names, where RNA and DNA nucleotides are given by adding the 5' monophosphate. So the letters A, G, C, U, and T are representing the bases, and they're often used in the abbreviations of the respective nucleosides and nucleotides. So you can see here in DNA we have A, G, C, and T, where in RNA it's A, G, C, and U. And then you can see their nucleoside names and their nucleotide names. All right, so let's do a quick little study check. So let's identify the following bases as a purine or a pyrimidine. So pause me and come back. Well, did you see that adenine is going to be that purine, right? Because it has the 2 circles, or the phosphate rings, right? Where the cytosine is going to be a pyrimidine because it only has the 1. All right, here's another question. Identify each of the following bases as a component of DNA, RNA, or both. Guanine, adenine, thymine, and uracil. Pause me and come back. All right, so guanine is in DNA and RNA. Adenine is in DNA and RNA. Thymine is that T, and that's going to be in DNA only, where uracil is the diva, and it's like, I'm only going to be in RNA only because I am too special for T's. Okay, so ATCG is in DNA, AUCG is in RNA. Great job, you guys.